My name is Steve Ora, and I conducted the following study with Dr. Joanna Cunha as the project research academic advisor. Immunogenic characterization of the GAG gene in HIV-1 subtypes prevalent in Brazil. The HIV genetic variability represents a major obstacle both to the control of infection by the host immune system and to the development of effective drugs and vaccines. CTLs, recognizing epitopes within the HIV GAG polyprotein, have been associated with early infection control. Besides, these epitopes are being used in current approaches for vaccine development. This study aimed to investigate the variability of HLA class I binding epitopes within the GAG gene of HIV-1 strains circulating in Brazil, retrieved from Los Alamos database. HLA-1 allele variability and predominance in Brazilian population were determined using the allele frequency net database, and epitopes within the GAG region were identified using the immune epitope database. The Swiss model program was used to model the structure of GAG-encoded proteins using the epitope with the best identity value. Protein epitopes recognized by the most frequent HLA-1 alleles in Brazil: HLA-2, A24, A3, A1, A68, B35, B44, B15, B51, and B07 were identified in the reference sequence HXB2 and in the most prevalent HIV-1 subtypes circulating in Brazil. 1,320 epitopes were found, 316 in HXB2, 336, 321, and 302 in consensus subtypes B, F1, and C respectively. 24 highly conserved candidate epitopes were found among subtypes. Important data found. The allele HLA-A2 is the most prevalent in Brazilian population. It is possible to notice that the consensus sequences present different quantities of epitopes. For example, the F1 consensus sequence presents a minor amount of epitopes. Probably that's due to the substitution of antigenic regions, therefore favoring the viral escape. The epitope described on screen wasn't listed in any academic or patented database, thus believed to be a novel promising epitope for vaccine purposes. Thus, the subtype-specific variability of HLA-1 binding epitopes within the GAG gene of virus strains circulating in Brazil described here may contribute to the characterization of HIV-1 immunogenicity, as well as to vaccine development studies. Acknowledgement. Federal University of Bahia, Multicenter Graduate Program in Biochemistry and Molecular Biology, Biochemistry Laboratory, Federal University of Bahia.